Hello, everybody. This is Chris McAdoo, Chief Experience Officer at the Knoxville Entrepreneur Center, a business accelerator located right in the heart of downtown Knoxville, Tennessee, dedicated to changing lives through entrepreneurship. Entrepreneurship, man, I just love talking about this stuff, y'all. I am stoked to have Jose and Alejandro Nuno with us. Jose, if you guys want to tell us a little bit about yourselves and a little bit about your company, Narval.
Okay. Let me start with myself. I'm Jose Luis Nuño. I'm one of the founders of Narval, CEO. I'm a scientist turned into business by chance and never came back. This is our third startup working together. I have been most of my career in biotech, involved in the development of vaccines, diagnostic tests, and now therapeutics. I'm married, I have 2 children, and fun fact is, one of my daughter's first word was virus. So that shows commitment with the cause.
Okay, so I'm Alejandro. I'm also a chemical engineer by background. I have mostly been involved in the technical part. We have been working together in 3 startups. So I have always been the product guy, developing the products and testing and everything. So that's it.
We have been working together for almost 17 years as founders.
Well, and not only that, but in your second startup, you actually had your wife and your other brother, correct?
Yeah. Yes, actually, since the first startup, we were working together. So it was the 2 of us, Rodrigo, our brother, and my wife, Laura. For 2 startups, we worked together as founders. And so it's... Yeah.
That's incredible. Well, tell me a little bit about where you guys are from. And I understand your father was a chemical engineer as well. So you guys come by this honest.
Yeah. So, we are from Guadalajara in Mexico. Again, we have been involved in biotech our whole life, but I think, as Alejandro had mentioned earlier, I think most of our interest for engineering and chemical engineering, chemistry, etc., it's because our father worked for his whole life at Petróleos Mexicanos, the national petroleum company in Mexico. So we lived our whole lives seeing tanks and pipings and stuff like this and distilling oil, etc. So I think at least at the beginning, the interest for chemistry came from our father.
Yeah, then we got interested each at their own time in biotech. So it was like a step-by-step road that led us here. So, I'm also chemical engineering, and as Jose said, we got primed to chemical engineering from our father. So after chemical engineering, we both got interested in biotech, and so each at their own time. And this has led us step by step until this point. We have been always working in the biotech area and different markets, but always biotech.
Well, let's talk a little bit about Narval. So antibodies. Tell me what the focus of your company is.
Okay. So at Narval, we are pioneering in a new class of antibody drugs that can be delivered through body tissues to treat diseases on eyes, lungs, and skin. So the problem that we're solving is that existing antibody drugs need to be delivered through the bloodstream at high doses to secure that the antibodies reach the target disease site. The problem is that this increases the risk of systemic side effects and, of course, the cost of the treatment. A good example of this will be, for example, COVID. The first antibody drug for COVID need to be injected into the bloodstream, and several times it created the risk of anaphylaxis and allergy reactions and stuff. And it will not make sense that if you have a respiratory disease to be injected with the antibody, but they cannot be used other ways. So what we do at Narval is we are developing a new class of synthetic proteins that mimic the activity of an antibody in order to be able to be delivered by mucosal or skin tissues. So we create these antibody mimetic proteins, or AMP for short, fusing in silico the CDR3 region from a shark antibody into a protein scaffold from a sea snail. And then we optimize the capabilities of these antibody-like proteins using machine learning and artificial intelligence algorithms. So these AMPs are 40 times smaller than a conventional monoclonal antibody used for antibody drugs. And of course, because of this, they can cross through tissue barriers on your body. So they can go in through your eyes, onto your lungs, or into your skin just by adding them in the topic, like a topical application. And they are heat resistant, which is very important because that way we can create pharmaceutical forms that were impossible to do with monoclonal antibodies. So we can have our antibody-like proteins in liquid or solid, for example, in pills, etc. And also very important, they are produced cell-free. So we don't need cell cultures or bioreactors to create these antibodies. So they are inexpensive to manufacture and very easy to scale up. So with that capabilities, we are able to create completely unconventional antibody drugs applications like antibody drugs in aerosol, in eye drops, or by transdermal delivery. And the mission here is to be able to create affordable, accessible, and easy to use antibody drugs without systemic side effects to increase access to life-saving therapeutics for millions of patients.
Wow. Well, and you're the product guy.
Yeah.
So talk to me about how you are developing this and your intention around, I guess, for one, going the full synthetic, right? So it seems like you're opening a lot of opportunities there that an antibody drug with a particular set of those side effects, like you mentioned anaphylaxis earlier, you know, how important has it been for you guys to take that route in terms of actually the product that you were developing, and what does that mean for the scalability of it?
Yeah. These molecules are developed by fusing, as Jose said, part of the shark antibody with a scaffold from a sea snail. So for this to happen, we have first to get these shark antibodies. So when we have a specific disease we want to target, we select a biomarker. This is a molecule or a substance, well, a molecule, protein, something that has a negative effect on your metabolism, for example. So what we do is that we take this biomarker and we run it's called panning, but it is just exposing this target to different versions of the shark antibody, so we can select the one that blocks the function of the biomarker. Sorry.
Okay.
So, well, I think I got a bit technical there, but
No, keep going.
okay. So once we have this shark antibody, we sequence. What I mean is that we define the amino acid sequence that forms that antibody, and then we take out the part that, like the sticky part of this antibody, and that's the part that we fuse to the snail scaffold. So once we have this is what we call AMPs, or antibody mimetic proteins. So this is where our product really starts. This is done in silico, in a computer, and then from this point, the work is done in computers using artificial intelligence, computational tools, and what we want to do is to make sure that this AMP is capable of binding to the target, and also to determine the force, the effectivity, the affinity, and once we are satisfied, we can make some changes to the structure, like change an amino acid to make it more sticky, let's say. And once we are satisfied with this, we're at the point when we can start testing. But before testing, we have to produce it. So traditionally, the way to produce a molecule like a protein or peptide or something like this, you insert the genetic code into a bacteria and then you produce it. That's the classic way of doing it. Like a bacteria, maybe an animal cell, insect cell, something like that. But we wanted to avoid this, and since we have a very small molecule, as Jose said, it's 40 times smaller than a traditional antibody, we are able to synthesize them using
Synthesis.
That means there's a very simple process, because it's not really simple. It is a very it's a marvel of engineering. There's a way to add amino acids that to assemble the chain of amino acids. So these AMPs can be synthesized outside of either a bacteria or a eukaryotic cell. These can decrease the risk of some of the side effects that come from traditional protein production. For example, the pyrogens. Do you say pyrogens in English?
No, I'm not sure if pyrogens, but yeah.
Okay, there are some molecules that can induce, for example, fever or other reactions that come from the body of the cell. So if you take out the cell, you don't get those toxins.
Okay.
So you don't have to purify them.
I am going to back it up for a second because I'm curious. I loved how you just got so excited about, like, this is a marvel of engineering. Like, we are living in a time where technology, knowledge, and the ability to create things, not only is that right in front of us, but even since, a year ago or 5 years ago, we can 10 times what we would have been able to learn. So it's like really exciting to think about. I'm going to back it up though, and I'm going to ask just for a definition, because a lot of people that may watch this, they may be familiar with these terms, but they also may not.
Yep.
And they may also hear like, sharks and sea snails, and there's a lot going on there. So tell me, give me a best definition that you have of what is an antibody.
Okay, an antibody is a protein that is generated by an immune system to block the activity of something. That means, for example, let's say a toxin, venom, something that triggers something in your metabolism. So when you block that capability, then you can modify what's happening on the metabolism. So you can stop a disease, or you can stop, for example, a signal inside of your body or something.
Gotcha.
So it's for blocking or neutralizing something in your body.
Awesome. So you guys right now are in the preclinical stages. Tell me a little bit about what the next, 12 months, 18, 24 months holds for Narval.
Okay, so the goal is to go into preclinical phase with our first lead candidate. That means the first drug that we are developing. So this first drug that we are developing is to treat a disease called diabetic retinopathy.
Okay.
This is blindness because of diabetes. So this affects like 27% of people with diabetes. It's a very, very, very big deal. Wow. And the current treatment for diabetic retinopathy is to be injected into the eye with a monoclonal antibody, which is it's just kind of scary. You think about when you see it, it's way worse. So it's really freaky to see someone to be injected in the eye. The problem with an injection like this is, of course, at long term, it could be dangerous because at some point it generates other problems like geographic atrophy, which is also kind of blindness. And the other thing is really expensive. So the drug by itself could be like $2,000 per dose. But the most important cost is the whole thing around the procedure, because you have to go to the hospital with a specialist. For most people, it's going to be difficult. So outside maybe of developing or developed country like the US, in several countries, this is going to be something very difficult to actually come by. So what we are developing is a way to do the same treatment, but with our AMP to block the same molecule, the same target, but using eye drops. So our AMP is going to be diluted in an eye drop, so you can take the treatment at home, for example, with the eye drops, and that's it, instead of being to get an injection in the eye. So it's going to modify completely the way the disease is going to be treated. We are moving into preclinical testing by the end of this year, as I mentioned, and we have the goal of being able to apply for an IND and going to the first test in humans by the end of next year. So that's one of the reasons we are here, one of the reasons we are fundraising. We're also trying to get in a partnership with an institution like the University of Tennessee, et cetera. So the goal is to be able to start those testing as soon as possible.
Well, and what has it meant for you guys to have come here to Knoxville, Tennessee, to our little entrepreneurial ecosystem, and to be part of the Techstars program?
Okay. I mean, in my case, one of the reasons that I wanted to come here is, first, this program here in Knoxville is focused on deep tech and science tech. So the companies that you have been interviewing, everyone is doing different things, but all have something in common is we are deeply innovating in something, in science, in hard tech. So the program is focused on those companies that want to change the world doing something like this. And we have partners in this program that actually can make a difference on what we're doing. So partners like, for us, the University of Tennessee. Here in Knoxville, for example, we have a hospital. We have sites for veterinary testing, for example. In Memphis, they do have the School of Medical Sciences, etc., and a hospital also for clinical trials. So that makes a perfect sense for us to be in an acceleration program where these kind of things are part of what we do every day. Of course, there are other partners like the Oak Ridge National Lab. For us, for example, that we are doing things on AI, it could be also a possibility, a good partnership with them to keep working on this. So for us, it's like the best place to go right now.
That's awesome. And if someone wants to learn more about Narval, where can they go?
They can go to our website. It's narvalbio.com.
Awesome. All right. Well, thank you so much for everything that you are doing, and thank you for being here in Knoxville, Tennessee.
Thank you.